So recently people have found that a lot of our normal tissues are mutated and they collect genetic mutations with age. So for example, our blood, skin, and esophagus are highly mutated over time. And of course, if you're older, you collect more and more of these mutations. Now, whether or not these mutations have an important function, whether or not they increase the risk of cancer is really not well understood. So what we decided to do is look in the context of liver disease and cirrhosis to see if we could find any mutations in the liver, and then ask whether or not those mutations do anything. So we did very, very deep sequencing of diseased liver tissues from my patients in the clinic, and what we found is that there are lots of mutations in genes that were related to cancer and in some genes that were not known to be related to cancer. And then we used sophisticated genetic techniques to try to prioritize which ones of those genes are important for tissue regeneration that might be applicable to these patients while they're getting liver damage. We found a number of genes that were really important to promote liver regeneration. And remarkably, many of the genes that we found in our study are not found in cancer, suggesting they don't have a function to promote cancer formation in the liver. So what we found is that when you modify these genes, they can promote the regeneration and fitness of liver cells within a chronic injured setting, but they don't necessarily grow the cells in a path towards becoming cancer. It may just be that there's independent pathways for regeneration and cancer. So therapeutically, what this could mean is that we may be able to thread the needle and find ways of promoting tissue healing in the liver without promoting the risk for cancer in the liver. Moreover, I think it's really important to understand this so we can use this information to predict who is going to get cancer and who may not get cancer. So this could be clinically important because it would help us diagnose cancer earlier.